Okay, so our first presenter this morning is Andrea Clarke-Spence, from Dr. Spence Balfour's Lab, or Balfour Spencer's Lab, pardon me. And the title of her presentation here is an assessment of the challenges and opportunities associated with water quality monitoring in East Rural St. Andrews, Jamaica, from an environmental perspective. So tell us all about it.
Thank you so much. Thank you everyone for joining us this morning. My research is centered around water quality monitoring in Jamaica, and I'll be looking at the challenges and the opportunities. The title of my research is Challenges Associated with Water Quality Monitoring in East Rural St. Andrews in the Environmental Perspective. Supervisor, as we mentioned, is that the Env Health, and we're working from the Applied Disaster and Emergency Studies. Just before I dive into my research, I'll just give you an idea of the country of which I speak. So the country I'm speaking of is Jamaica, which is this country right here. Jamaica is the third largest island in the Caribbean. It has a population of approximately 3 million. It has a rugged terrain, topography with high elevation in the east. Those are the mountains. And there are numerous rivers and streams that are distinct from these mountains. Jamaica is also known to have a tropical climate. We have an average temperature of 24 degrees C, nothing like we have here. And we also have the seasonal rainfall, which varies from the wet season to the dry season. By the wet season, we are talking about from May to November, or dry season from December to April. Let me also define what is water quality monitoring. Water quality monitoring involves the measurements of water characteristics, including the physical, the biological, the biochemical, the radiological, and the microbiological parameters of water. Why is water quality monitoring important? Well, water quality monitoring is important for several reasons. The first reason I will highlight is for public health. The consumption of contaminated water, of course, can lead to numerous waterborne illnesses, which is debilitating for some persons, especially for our young and our elders, or persons who are immunocompromised. We have to ensure that the water that is being consumed is safe, and it will not cause any health issues for our population. It's important for adherence to law. Water quality monitoring is important as it satisfies local and international standards. We have different bodies that pay special attention to water quality, such as the World Health Organization, the United Nations, and this has always been an important issue for the United Nations, which of course made a move in 2010 to say that water is considered as a human right. So every human should have access to clean and safe water. As it relates to climate resilience, it's important to know the quality of water that is present in a country, the levels how it is impacted by different weather conditions and different weather events. And so it helps to prepare a country in terms of its climate resilience. It's important for policy making. The water quality monitoring also helps to inform policy making in terms of your land use, in terms of water resource management. And of course, our natural resources need safe water, need water as well, fresh water that will, of course, contribute to its sustainability. So that is very important that we also monitor those aspects. Let's give you a background to the research. In 2017, the Planning Institute of Jamaica conducted a survey of living conditions, and it found that 44.1% of communities and 78% of urban communities were using water from unimproved water sources. So these are sources that were not treated and were not monitored. In 2022, another research was done by Texas University, where it found that 100% of microbial water samples that were collected from roadside improvement tested above the national standards and above the levels which are required for total coliform. So that was indicating that there was pervasive contamination in these non-municipal water supplies. So these are supplies that were not treated and were not monitored, but they were still being used by community members. In 2023, the Kingston and St. Andrew Health Department also had readings for rural areas which were high above the national water standards. So the national water standards stipulate that for your total samples, you should be having at least 5% residual chlorine negativity and no more. And for your positive bacteriological, you should only be having 5% positivity. And as you can see from this this table here, you see for the negative chlorine, residual chlorine, all of these are way above the 5%. And for the positive bacteriological samples, all of those are all above 5%. So there's definitely a need for such a research. My primary research question is, what are the challenges and opportunities associated with water quality monitoring in East Rural St. Andrew, Jamaica in the context of sustainable environmental protection and human health? My secondary research questions are, what is the relevance of water quality monitoring in environmental protection and human health? What are the environmental factors impacting water quality in East Rural St. Andrew? And what are the challenges and opportunities which are related to water quality monitoring in East Rural St. Andrew? So from my history review, my hypothesis is environmental challenges negatively impact water quality monitoring in East Rural St. Andrew. What's the significance of this research? Now, this research hopes to highlight disparity in water quality supplied to urban areas in comparison to rural areas. It also hopes to highlight the population at risk for waterborne transmission. Also hopes to highlight the impact of the waterborne transmission to take place on the health sector and the country at large, and the environmental factors impacting water quality in the area. It seeks to also find viable opportunities for improving water quality monitoring and also information which is necessary for policy making. This is a map of my study area. This is St. Andrew right there. So we're looking at East Rural St. Andrew, this St. Andrew in Jamaica, and this is East Rural St. Andrew in the parish of St. Andrew. And this is also a better picture that shows you the communities that are in St. Andrew. Now, East Rural St. Andrew was chosen for different reasons. One, because of its location, you have mountainous areas and you also have coastal areas. And of course, this also dictates the population settlement patterns. You have more persons living in the coastal areas and you have less persons living in hilly areas. And these tend to be farmers and so on. So as it relates to access, for water quality monitoring, it's harder to access the hilly areas due to soil erosion, due to the topography sometimes. And of course, the population will also dictate the difference in monitoring needs. The more persons living in that area, the more you will need to monitor, and less persons living in that area, the less you need to monitor. And this is stipulated by the World Health Organization. There are also environmental factors of concern, as I mentioned, soil erosion, deforestation, agricultural runoff, and contamination by human activities. It could be recreation activities, domestic activities, or even water use for irrigation. So the area of East Rural St. Andrew has access to municipal supply, but it's limited access that tends not to be consistent. And so the members are forced to use water from rivers, springs, rainfall harvesting, and of course, these are not being monitored by any authority at all. So these are some of the alternative sources that they may use. This is a river in Dallas, the Dallas St. Andrew. Now this river is called the Miami River, and in Hood Bay is another river called the Cane River. Here you can see the level is kind of low, but of course I said that we have fluctuation as it relates to rainfall. So that's one of the reasons. Sometimes you'll have high levels, sometimes you'll have low levels. And in Mavis Bank and Content Gap, these are tributaries from the Hope River and from the Dallas River. And these are utilized when there is no access to the municipal supply. As it relates to my methodology, I'll be using a mixed methodology of quantitative and qualitative methods. For my quantitative methods, I'll be doing water testing, as I mentioned earlier, in the dry season and in the wet season, because we want to see if there's any difference in the quality as it relates to that. And there will be samples that are collected and taken to the lab for microbiological and chemical testing. For my qualitative method, I'm using semi-structured interviews and observation guide, and this will be for my regulatory agencies. My study area, my water sampling sites, these were also chosen purposely because it had to satisfy a set of predetermined criteria. For example, I wanted an area that has limited access and an area that also had access to other sources. We also wanted areas that had no issues in terms of environmental factors affecting the water quality, and so those were purposely. My data will be collected and it will be analyzed using SPSS and Microsoft Excel. There will be an integration of what is happening in real life and what is the perception of some of our regulators versus what is really there. Of course, there will be human subjects, so we have to take ethical considerations as well. The research will ensure that there is respect, there is protection and fairness to the participants. It will focus on consent, confidentiality, so safety and transparency. The research will ensure that it satisfies international ethical standards as well as the ethical standards of London University. At this point, I am at the stage of preparing my submission for ethical consideration. And as it relates to limitations, water samples may be impacted by conditions and accessibility of site because the area is known to have landslides, there may be soil erosion, or we don't know what will be happening in terms of our weather patterns at that time. And the interview responses may reflect personal and organizational biases. Budget or time constraints may limit our water sample. And in summary, water quality monitoring is essential for the area, although it faces numerous environmental challenges. This study will provide well-needed assistance in strengthening monitoring systems through environmental awareness, and these findings will support sustainable water resource management. And I want to extend special thanks to my supervisor, Dr. Clarke-Spence, the Kingston and St Department, the National Water Commission, so water provider, and of course, the economy. Thank you.
Thank you very much. Yeah, let's do here. Hopefully there's questions from the audience. Okay, so I might start with the question if people a few minutes here. Did you have one? Okay, okay, go for it.
Yeah, thank you. Is obviously very important work. How often are you actually physically going back to Jamaica to conduct this research and then coming back here? Oh, really? Okay. How often are you going?
I'll be going twice.
Twice.
Yes. So I'm doing in the wet season and the dry season.
Okay.
Go somewhere.
Is that for getting a wider range of
Yes.
data?
Right. So we want to compare the changes in terms of the readings that you'll get in the wet season, that's the rainy season, versus those that you're getting in the dry season.
Thank you.
I know you said that it's primarily rural, but is there any sort of larger urban centers like in this area, or is it just like?
No, St. does have urban centers. I'm paying attention to the rural areas. There's the urban centers which are more populated, tend not to have so many issues. Well, first, they don't have that kind of topography. So in terms of environmental factors, they're not so much impacted. And another thing is that there is way more monitoring that is done when you have added population, as is stipulated by the World Health Organization.
So, yeah, I'm just curious. So once you've gotten your results, so what's the next step like? You have to go back to the committee to implement such results or solutions.
Well, this information will be shared with the health department and the Ministry of Health. So they want to have intervention in terms of any kind of new systems that they want to put in place, and this information will be listed to them. But for me primarily, yes, I would want to, of course, let the community know what are the findings that we are having. And of course, we could also help the Ministry of Health in terms of putting out, developing advisory and so on to help them to be doing their own monitoring as well as to do interventions that can actually do some kind of treatment that will make it safe for them.
Okay, thank you very much. Thank you. Okay, our next presenter this morning is Rochelle. So Rochelle is a bachelor student with Dr. Etsuko Yasui. The title of Rochelle's project is Exploring Disaster Risk Management within the Agricultural Sector in Nigeria. So tell us all about it.
Thank you so much. Good morning, everyone. My name is Edward, and my name is Edward, and I'm currently pursuing Masters of Environmental Lifestyle at Brandon University under the supervision of Dr. Yasui. The title of my proposed research topic is Exploring Disaster Management within the Agricultural Sector. Introduction. Flooding occurs when excessive rainfall or water exceeds the large capacity to absorb or drain water. According to NEMA, Nigeria's food security increasing at risk as floods sweep through Nigeria states. In 2012, Nigeria experienced its worst flooding in over 40 years because the heavy rainfall across the country and the incidents affected 52 states in the country. In 2024, Nigeria faced a devastating flood affecting 1.4 million hectares of land, and economic loss from the disaster totaled around $7 billion, according to figures from National Emergency Management Authority (NEMA) and Food and Agricultural Organization (FAO). In 2025, NEMA says flood in the first half of 2025 saw 9,250 hectares on land in 47 local governments and 20 states. Now to my study area. Nigeria is a country in West Africa, and it covers an area of 9,023,709 kilometers, with a population of more than 236 million. It is the most populous country in Africa and the sixth most populous in the world. Nigeria comprises of 36 states and establishes Africa. So this is Nigeria on the map. To my study area, Nigeria's agricultural sector forms the backbone of the economy, employing over a third of the population. Focus. The reason why I focus on this particular region because it's been affected by flood, which is the native base, Niger Basin, and Northeast. And the reason for the selection is because of high disaster frequency to seize the diverse livestock and across the state. My research objective, the purpose of this research is to analyze the structures, challenges, and opportunities in disaster risk management. So this is the reason why I'm assessing the existing disaster risk management institutions, identifying major disaster threats and their impact on agriculture, examining community vulnerabilities and adaptive strategies capacities, exploring the role of policy and financing in disaster risk management, and then to recommend sustainable adaptive capacities. My research question: one, what are the common disaster risk management methods in agricultural products? Two, how do different types of disaster affect agricultural products and livelihood? Three, what are the main barriers to effective disaster risk management? And four, what policy reform and financing mechanisms can enhance adaptive capacity strategies? The significance of the study. Why does it matter? Agriculture is the backbone of the economy, and it helps reduce poverty and food security because of these farmers are small-scale farmers. And once disaster happens, it destroys everything which affects their crop, their farm, and their income. Contribution to SDG 2, zero hunger, SDG 2, zero hunger, and SDG 3, climate action. The research will help Nigeria and to also provide a safer and healthier future. 3, to provide evidence-based insight for researchers, policymakers, NGOs to design more adaptive strategies. The methodology. I'm going to use a mixed method. That means I'll be combining numbers and experience. And then for my data sources, I'll be using secondary data. That means I'll be using data that's already been collected by organizations like farmers, food and agriculture organization, National Emergency Management Authority, and World Bank. Then the primary data will involve household surveys, which means asking questions from farmers who have experienced disasters, interviews with key informers such as NGOs who are experts in disaster, and then case studies from past disasters, looking at specific disasters that has happened in the past, and know when it happened, what has been done, and what can also be done after it. The focus group discussion with farmers. Listen to the stories of farmers, their challenges. The analysis, because I'm using a mixed method approach, I will use different analysis tools. SPSS, which is a computer program, I'll use it to analyze numeric data from surveys in order to identify trends, patterns, and relationships. Then semantic coding, I will use this for interviews and discussion. Moving the ideas and see common themes. Summary. In summary, this research seeks to evaluate the effectiveness of existing disaster management structures in Nigeria's agricultural sector and to generate evidence-based recommendations that enhance adaptive strategies, safeguard food systems, and contribute to broader national and global sustainability objectives. Acknowledgement. I extend my gratitude to Supervisor Dr. Yashmi Suko for support and guidance, and special thanks to key participants, agricultural agency, and local farmers who will contribute to this trend.
Thank you very much for that. Any questions?
Go in the right direction.
I'll ask the same question as Carson then. How often are you going back to Nigeria to do the field work, or will you just go back for like the whole summer? What's the?
Yeah, the plan is to go back to Nigeria next month.
And so just one field season then?
Session, it's information and getting works.
If people aren't take here, so you're talking about doing like mixed methods and interviews. So can you give us a sense of like what types of questions that you'll be asking? I know that you can't ask leading questions, but like how do you plan on like structuring these? Give us a little bit more detail on that.
I feel questions all has to take about this.
But once you have that, or are you really waiting on that ethical permission to even start planning the question?
Infrastructure in place already for preventing floods, or is it kind of?
Yeah, government infrastructures.
Sorry, I'm gonna ask another one here. So with the flooding, is this from, just kind of total rainfall, or is this more in terms of rivers overflowing their banks?
So it depends, sometimes heavy rainfall continuously for this, for the reason for the cost. And sometimes the one that happened in 2020 was also the dam, there's a border between Cameroon. So there was a flood from Cameroon to the Nigerian dam, and then spring for the dam.
Kind of a complicated question. Any other questions? So another time.
Do you foresee any challenges occurring, like, what do you think would be the biggest challenge of like collecting your data, beginning your research?
The biggest challenge is relating with the farmers, because I need to speak with them in a local language because most of them are not going to speak. No, that's fine. And if I want to collect something, I need to translate it to local language when I'm trying to collect it. But some of them are not going to speak. So I can say that is the nature.
Thank you very much.
Any other questions? Okay, seeing none, thank you very much. Okay, our next presenter this morning is Afsana, who I believe is also with Dr. Masui. Afsana's presentation title today is Socio-Spatial Vulnerabilities and Resilience in Flood-Prone Communities in Bangladesh, sensing a flooding peak. So tell us all about it.
Thank you so much for the intro, and welcome everyone for this special event. So today I would like to talk about my thesis proposal. So moving to my title. So my title is Assessing Transformative Water Space and Dam Use Management through Remote Sensing Implications for Social Spatial Vulnerability and Community Resilience in Town. And my supervisor is Dr. Itsuko Yasui. So at first, I would like to talk about some background of my study. So Bangladesh is a South Asian country and it is located in a rural area. So for its geographical location, Bangladesh is surrounded by a lot of regions. And for that, Bangladesh has to face a lot of natural disasters every year. And who knows, flash floods, cyclones, and river migration, river embankment. These are the most common phenomena where people of Bangladesh are dealing with in every year. So most studies focus on physical factors like rainfall, elevation, and drainage density that are the most common factor for these disasters. But when I go through the literature, so people have created like a lot of create some controls in order to flood. They created dams, embankments, and sometimes these dams create upstream blockage and often triggers flash flood even more. And for that reason, downstream areas face water scarcity or drought condition. So here I am representing the real scenario when flood in Bangladesh. Then this is the real scenario happening in lowland area like Bangladesh. There is no dry land where people can move and people can do their daily work. Everything is underwater. And moving to my second picture, this is another scenario when in the rainy season there is a scare of the water, which is important for the agricultural ecosystems and basically it's a drought condition. So why I am interested in doing this research? As I said, when I go through the lot of literature, I found some gaps that people are more interested that this disaster happened most likely for that natural way like drainage density or elevation model. But when I go through, I saw some gaps that mismanagement of dam controls, and these dams in Bangladesh were built for irrigation and flood control, but suffer from weak and outdated management. And poorly planned or agent structure often intensify upstream flash flood and create downstream drought conditions. So these issues disrupt farming and damage ecosystem and affect community livelihoods are very good. So this research will combine spatial and environmental data on these dual impacts, which is still missing. And this study will examine how dam needs management and drives both extreme and outline sustainable water management options. So moving to my study area, here you can see Bangladesh, and India, the surrounding India, the neighbor country. So I am interested in doing my research in this area, the northern part of Bangladesh, right here. So if you move to the real study area, my research will fall that with this in these three cities, which is the major three cities in the northern part of Bangladesh. Here you can see the bottom is the Bangladesh, and these three cities is consisted of Lalmonirhat, Nilphamari, and Rangpur. These three cities are the major three cities which is situated in northern part of Bangladesh, and there is a major river crossing over these three cities, which is the Tista River, and this river is the main important thing, and this is called the life of the people of northern people and Indian people as well. So for my research objective, my first objective is to examine how land use planning, governance, and transboundary water management influence disaster risk reduction and recovery in northern part of Bangladesh. And the second one is to utilize GIS and remote sensing to assess vulnerability and resilience in communities along the Tista River basin. And my third one is to develop community-based disaster risk reduction to Tista affected region to promote sustainable and climate resilient development. So for my research question, my first question would be, how does Tista River's transboundary management affect flood and drought patterns in northern part of Bangladesh? And my second question is, what are the key social spatial vulnerability zones identified through GIS and remote sensing? And third one is, in what ways will local governance and land use planning affect disaster preparedness? And lastly, I will try to find out how can community-based strategies strengthen resilience in Tista basin community. So for the methodology, I decided to do my methodology in three parts. Like first, I will do some data collection. So for this, I will heavily depend on satellite data. And here I basically do Sentinel-2. I basically use Sentinel-2 and Landsat dataset. And I will go through some hydrological reviews. Here I will review some previous flood events in my country. And I will do some community survey as well. And lastly, I will go some existing policy reviews over here in order to identify the data. And lastly, secondly, I will do some GIS and spatial analysis. Here, I will do some GIS mapping of temporal land use and water flow analysis through GIS analysis. And I try to identify drought hotspot and model dam impacts of the local area of Bangladesh, and a model scenario showing impacts of dam operation on downstream areas. And lastly, I will do some qualitative analysis. Here, I will overlap all of my findings here and try to do some applied thematic coding, to interview the local people and focus with group cases. And lastly, I will try to identify patterns in community adaptation, governance gaps, and local resilience. So here I want to clarify my last methodology, what is thematic coding. So thematic coding basically a qualitative analysis method used to identify patterns to introduce people and focus group data. And it involves reading participants to response carefully and assigning short levels to important statements and grouping similar codes into broader themes. And lastly, it will help to using these themes to extend community exposure, challenges, and resilience. So in simple way, thematic coding in this study, thematic coding will help identify how flood-prone communities adapt to water-related hazards and what barriers they face and which governance gaps affect their resilience. So through this conducting this research, I am expecting some outcome as well. So if I am able to do this research successfully, then I can identify flood and drought hotspots in the Tista River, which is the northern part of Bangladesh. And I will do through GIS mapping, I can suggest some mapping impacts of dam operation on local communities. And I also can reveal social and governance factors in vulnerability. And it will also help to highlight community adaptation and resilience patterns. And lastly, this research will help to recommend sustainable water solutions in the northern part of Bangladesh. So every research has some limitations as well. My research can face some limitations as well. As I said, my study area is the very near in the India-Bangladesh border. So I may have some problem to access some high level data for the political issues, maybe. So limited access to upstream data and time and resource constraints can be a big problem for my field survey. And my research is heavily dependent on satellite images. So cloud coverage and image resolution can be a big problem for me. And rapid land use changes may affect the temporary accuracy. Because in this northern part here is some river banking pavements. This is a common scenario of that part of Bangladesh. So I can find some detailed accuracy on that part. So for conclusion, this research addressed how dam management shapes flood and drought in Bangladesh, and this will help to combine GIS and field data and governance to analyze the signal. And this research will support community-based and data-driven disaster planning as well. And lastly, it will offer insight for sustainable transferable water management. So, lastly, I want to acknowledge my professor, Dr. Suku Yasvi, who gonna help me to promote my research and along this organization where I can find my primary data as well. Thank you everyone for your questions.
Okay, hopefully we have lots of questions. Please pass them back.
I was just wondering if you could expand a little bit on what you mean by dam mismanagement.
Dam mismanagement actually, as I said, the people of Bangladesh, Bangladesh is a lowland area. So if the flash flood is a common scenario in every year, so in order to control the flood, people have built some dam, and sometimes this dam does not work properly. So then this situation sometimes causes the drought condition in the downstream areas. So yeah, this is known as the dam mismanagement because people are not able to create the dam properly. So yeah.
Will you be going to Bangladesh to provide aerial data?
Yeah.
Okay, so that's in addition to the.
As I said, in the field survey, I will interview the local people to understand the real scenario, what is happening on the northern part. Then I will compare those scenarios to my previous flood events and mostly the comparable data collection.
Does the government have like online or real-time hydrometric data, like stream gauging data? Like in Canada, we have rivers and streams all over the place that you can get real-time flow records from.
So I said the flood forecasting warning center, these organizations have some real-time data, what type of rainfall is falling, and they have the real-time data.
Yeah, so you could probably generate some rainfall runoff ratios and see how they're changing through time with changing land use and stuff. Like there's lots of low-hanging fruit if those data exist that you could do some analysis on as well to inform some of your research objectives.
For your land cover data, what are some of the types of data land cover are looking at? Is it buildings, vegetation?
Yeah, it's a different because Bangladesh is a developing country, so it's hard to find any urban it's rural area in Bangladesh because Bangladesh is developed and they create different kind of structures over there. So I will try to find those why flood is happening over there because there is no any kind of wetland rural areas because people are creating this infrastructure over and over. So yeah, I will try to find those.
So in your communication, you mentioned limitation due to satellite coverage as a result of flood. So in your analysis, how do you take care of this limitation? Because it's my skill your data perhaps my not get the expected results.
Yeah, further, as I said, my data is heavily dependent on GIS analysis. So yeah, that's why I will use satellite images, the satellite Sentinel-2 images, because it will give us my more resolution and more accuracy for the land coverage and any kind of cloud coverage. So I try to use landscape data and Sentinel-2 data, and it will help me to build up those accuracy. because of the cloud coverage is not clear for most of that. So I will try to collect data in rainy season. So there will probably a lot of rain on that time period. So it will be a big problem for me. So that's why Sentinel-2 will be help me to analyze that first.
Thank you. And again, why are you so much centered in northern Bangladesh? So why not other region?
As I said, the river Tista is the main important for the people of Bangladesh and neighbor country India as well. And people in country Bangladesh have created a lot of dams over here. So if I want to clarify the transboundary water basin, so that's the best for me. So there is the issues over here. The people are facing drought conditions over the year, and people are fighting for their right to water to give the right water. So that's why I choose that.
We have time for one last question.
Kind of two, I guess, so one. It'd be kind of cool, like, I don't know anything about this, and this might be out of the scope of your project, but like, if you compared like, because you're comparing management, right? So you kind of do a baseline, so it'd be cool to kind of do kind of dam like, what are you using for your management baseline dam? Like, there's a dam north of here, 2 hours, the Lake of Prairies, right? And that controls the water that goes to Grand. That was originally built to hold back water for the farmers in case of drought. But then, of course, during flooding, now that occurs, Brand, they hold it back a little bit, like all the time. But they still kind of sometimes mismanage that, and that's predictable. So it'd be kind of cool if you could compare that here to there, and then seeing like first predictable versus the method conditions in Bangladesh are more unpredictable. So you have a baseline management of what good management looks like. And then, it's not much of a question. We're just idea. It might be out of the scope of your project.
Actually, this is in my preliminary stage. I will find out some literature reviews, and when I go to the field survey, then I will understand the real scenario. What is happening. So these actually a literature review, I found those things, and most importantly, I will do this through GIS and remote sensing projects. And these things are the main for my research and when I go through. So this is the main thing for my research. I will do everything through GIS and remote sensing. So that's the thing.
Okay, thank you very much. Okay, so the next presenter this morning is Danielle Stolt from my own lab. Sadly, Danielle is standing between you and lunch. So Danielle is going to talk to us about an emerging story in my lab, which is a little bit new stuff and direction for us. So her presentation is meant to be cooperative and organelles talk how cytomyal communication is disrupted in bone rising.
Thank you, Dr. Schmidt. So ribosomopathies are a class of rare genetic disorders that either directly impact the ribosome itself or more indirectly is how the ribosome is built and sent. Which leads you to your first question, well, what is a ribosome? And in the most simplest terms, it's a complex molecular machine, and it's responsible for building all of the proteins that sustain and maintain our life and all of our function. So back to ribosomopathies, historically, we've looked at these more narrow and just looking at how they affect the ribosome, where we haven't stepped back to take a more whole cell approach to look at this. Which is important because we know that ribosomopathies often lead to early onset cancer progressions. But this is not explained by just looking at how the ribosome assembles or how the ribosome is working. And in order for us to take a step back and look at the whole cell, we need to first understand different parts of the cell and how we can categorize cells. And all your favorite friends, all your favorite organisms and plants and bacteria who tend to have one will fit into one of these categories of the basic building blocks of life for the cell. And these two categories are the eukaryotic cell and the prokaryotic cell. The eukaryotic cell, eu meaning true, and here we're talking about the formation of membrane-bound organelles. We're just small compartments within the cell that are known to have a specialized function that we often conceptualize as independent or individual. They also have the presence of this cytoribosome, termed cyto because it resides within this empty space or the cytosol. On the prokaryotic side, pro meaning before, we have before membrane-bound organelles, so therefore they lack membrane-bound organelles, but also have a little bit different of a ribosome. It's just that BS ribosome doesn't really matter, just know that it's a little bit different.
And where this becomes interesting is if we actually start to tease apart the eukaryotic cell and look at some of these membrane bound organelles, specifically the mitochondria. And we note that it kind of comes with its own set of ribosomes and translation products. And it can make its own stuff and its own genetic material, similar to how prokaryotes do. And we see the rise of the mitoribosome, mito meaning it comes from mitochondria. Also, what's interesting in understanding some of the concepts that I'm going to share today, this is a double membrane structure, which we call the outer and the inner membrane of this structure. And so the question that we have to ask ourselves is why? Why do these two independent systems choose to reside within one another? And evolution has done a really good job at explaining this, but you don't have to understand it from an evolutionary perspective. As today, we're just going to look at it from a metabolic perspective. So the eukaryotic cell by itself has two choices when it comes in contact with a sugar molecule, what you might know as blood sugar, but I'm looking at it at a microscopic scale. But the first choice being to go through a process of fermentation to get some cellular energy. Second choice, which is a little bit more expensive, is to further convert the sugar into more metabolic products. That can then be shuttled to the mitochondria if we have one. It's important to note, and this will be a recurring theme within my talk, that you can actually shut down this process of fermentation in a majority of cells and force the cell to rely on the mitochondria that it has. So we can measure certain things like what the mitochondria is doing. And well, what is the mitochondria doing? Mitochondria brings forth the ability to do more complex metabolic processes with these metabolic intermediates, one being oxidative phosphorylation, or what I'll call OXPHOS. This occurs between those two membrane structures, which I already talked about, the outer and the inner membrane, and consists of 5 protein subcomplexes. 4, cleverly labeled 1 through 4, like biochemists tend to do. And the last one, ATP synthase, which is responsible for making all of ATP, or the energy of the stuff. So you can think about this at a microscopic level, like your heartbeat and metabolic processes, or at the bigger level, like how you want to run or jump or whatever else you want to do with it. And so, with all the information that I've provided you so far, knowing that the mitochondria comes with all its own pieces, it might be tempting to assume that the mitochondria is then responsible for producing all of these protein complexes that it requires to do complex metabolism and OXPHOS, which actually is not the case. A lot of these complexes come from the nucleus, or the cytosol, or the cytoribosome, which you might be more familiar with. And actually, it turns out that at the translation level, the level of cytosomes, this process is unidirectional, meaning what happens in the cytoribosome likely depicts what's going to happen in the mitoribosome, and the mitoribosome might try and kind of balance itself cytoribosome, depending on what it's doing. So to summarize some of the literature that currently already exists within the field, although go into more depth in just a moment, and look at this by looking at how the cell uses these mitochondrial complexes and how it adjusts itself to the cytoribosome. And again, we're going to shut down fermentation, and we notice that the mitochondria in the mitoribosome will upregulate itself, just meaning that it's going to provide more components and therefore more efficiently produce this energy, because it's now the sole source of energy. However, if we shut down the cytoribosome, that's when we see things kind of start to go a little bit crazy, and we see that it induces some mitochondrial stress, some overall cell stress. We can see cell death or cell destruction, which we often call apoptosis. These pathways are all commonly known to lead to cancer progression, which we do see within rhizomopathies, which is why it's been a hot topic in our lab. So with all of that, we already know what happens when you completely turn down the cytoribosome and shut it off. We know what happens when the cytoribosome is fully functional, but what we don't know is what exists somewhere between these, and not fully shut off and not fully turned on, and that's where the rhizomopathies lay, which is a focus in the Shred rhizomopathy lab. Specifically, within our model organism, we work on both Rett syndrome, or PCS, which can be used as a representative model, although I'm hoping that my work will reach a broader spectrum and being applied to rhizomopathies as a whole, as we know that this cancer progression comes from a lot. So throughout my master's, I have 3 main points that I'd like to touch on today to tell you about some of the work I will be doing. First being a literature review, and I hope at this point I've at least proved that I've done some of my homework. Next, I'll be moving into ATP assays, which I can use to measure the production of oxidative phosphorylation directly. And lastly, I'll spend a lot of time doing a whole lot of medical science, but I'll share some of the limitations that are going to direct the trajectory of my master's and what those will look like in the long term. For my literature review, I just want to reiterate that this idea of cyto-mito-communication, or the talking between the cytoribosome and mitoribosome, is not a novel concept. It's actually been extensively researched by Dr. Sterling Churchman, and a lot of my references are coming from her lab. She's in the genetics department at Harvard Medical School. And this is actually a really beautiful graphical abstract that comes from one of her papers, where it might look a little bit scary at first, but if I actually show you one of the diagrams that I've already presented and talked about, we can see that it's not so scary and it's just the same complex, and it's just depicted a little bit differently. Dr. Sterling Churchman has chose to show specifically which parts of each complex have come from the mitochondria, shown in that red or orange, and which pieces come from the nucleus, shown in the blue, and you can see how this would be a coordinated process. She also focuses on different factors like production and abundance of these complexes and how they start to try to balance each other. So I know that the gap that I'm trying to fill is how ribosomopathies come into play, and how ribosomopathies will affect this communication path. This has been a little bit of a challenge since I've worked in the lab for a few years now. It's taking some of my previous knowledge and specialization in ribosomopathies and bone neurotic syndrome, but also taking in the new information with the cyto-mito-communication story and trying to kind of feed these pieces together to make a little bit of a story and a hypothesis. But overall, I'm hoping that all of these concepts are going to come together to help us understand how ribosomopathies can lead to cancer and early onset cancer type risk. Since I'm in the Masters of Environmental Life Science program at Brennan University, my study is multidisciplinary, which is going to bring in some genetics, since a lot of my literature review has come from the genetics department at Harvard Medical School, but also bioinformatics, since a lot of the basis for my hypotheses have come from proteomic and soon, hopefully, transcriptomic data, which shows some different regulation of mitochondrial genes. I will also be taking some metabolomic and cellular energetics, which we'll see in just a moment, but this is just the quantification of ATP and some of those metabolic intermediates, which I already talked about. Of course, since I work in the biochemistry lab, I'll also be employing some of our classic biochemistry and molecular cell biology techniques in my experimentation. And lastly, I'm hoping that my work will extend to medicine and pediatrics as I'm trying to bridge this gap and understand how cancer comes from ribosomopathies. Also coming from my literature review, and the second step of my master's thesis, is looking at ATP assays under the varying cell conditions, which has led me to about 3 to 4 months of protocol development. First, in different solutions like medias and buffers, and how we grow cells and how we can treat the media differently to induce different states within the cells to start to study some of these processes. Some of these states being mitochondrial disruption, or just completely shutting down the mitochondria and forcing the cell to rely on fermentation. Secondly, we can look at the experimental inhibition of fermentation and oppositely force the cell to rely on the mitochondria. This has already been done in literature, but we're working on bringing it into the ribosomopathy lab since it's new to us here. And lastly, ATP quantification assays are hopefully where I'll end up for my preliminary work, just so that I can measure how oxidative phosphorylation is working as a whole to understand how the ribosomopathy is affecting this complex metabolism. So that brings, and that's where I am about now, but ask me again in about a week and I'm sure it'll change. So that brings me to some of the limitations that are going to affect the trajectory of my master's, since this is all preliminary. But it's important to understand that since our hypotheses are somewhat novel, that we're often underrepresented within scientific literature, and we don't have a lot of literature to rely on to support our hypotheses. For example, these are often ribosomopathies underrepresented in clinical settings. Bone neurotic syndrome specifically is present in about 1 in 355 birds exclusive to the Hutterite population, which means it's also almost completely exclusive to the Canadian Prairie provinces, and the Charette ribosomopathy lab is currently the only lab working on bone neurotic syndrome. This means that we see the same underrepresentation within scientific literature, meaning that we often lack support with our novel hypotheses. So with that being one major limitation, another one is that we also lack limited human cell models, and therefore our model organism of choice has been yeast, which has highly conserved metabolic pathways, and it is somewhat representative, but it's important that once we get this preliminary data and we kind of have an understanding of what's going on, that when we move into human cell models or just actual human cells, that we're able to ensure what we're doing is meaningful and we're making good use of these cell lines. Of course, I've also been stuck in protocol development, and sometimes you can read as many papers as you want and write as many recipes as you want, and as soon as you try to do it, it's not going to work, which means I've had some failed attempts, but I've also had some successes so far. And like I like to talk about in all my thesis presentations, time is of course a limitation as I move forward, and I'm already starting my 4th month of my master's project. So to summarize, since I don't exactly know what these 2 years will entail, I just wanted to re-highlight some of my goals and what I'm hoping to accomplish with my master's. One being understanding this pathway of ribosomopathies and how they affect the cell as a whole rather than just the ribosome. Specifically, I'm focusing on the cytomicro-communication of the ribosomes, and I'm using ribosome syndrome as a representative model of ribosomopathies. I'm hoping to fill these gaps within the literature of cytomicro-communication and what happens when the ribosome isn't necessarily turned off or fully functional, but somewhere in between in ribosomopathies. And ultimately, I'm hoping to resolve possible mechanisms that lead from ribosomopathy to early cancer progression in patients. With that, I have to thank, of course, Dr. Mintel-Schredt, who has supervised me for the last 3 years and will continue to supervise me through my master's and overlap me, but also in the Gemini Lab, who have been so kind in bringing their materials and some equipment to us as we've started some of these assays. I'll share my references, and thank you. I'll take questions.
Thank you, Danielle. I'm hoping there'll be lots of questions.
Can you go over again, like, so what are the states that you want to look at and compare? So like start off fermentation, or if you want to just go to that, I'd like to hear.
So because
Go back to the slide if it helps you.
Here. So because these models just a little bit different than humans, they actually rely on fermentation, whereas human cells don't really do that. So what kind of experimentally can be done is we can shut down the process. It'll mimic more of what human cells do and forcing some lines from the mitochondria. So that's where that one comes from. And then for shut down the mitochondria, that's more to check that our medias are actually going to shut down fermentation, and it should be able to reduce cell death for just completely shutting down the metabolism. And then we're hoping to do these HPV assays as preliminary work, just to kind of assess how osmosis is working as a whole before we start looking at things like oxidative stress or like protein of compounds. So then, like with your models, you would be comparing the bone one with the bone one in both.
You mentioned that you kind of change your perspective, like looking at cell perspective. I'm just curious, what initially got you in that direction? What about your project?
So kind of in my last few years, I've been everywhere within ribosomopathies. I've kind of done like specific abundance of like what is causing syndrome, but I've also done just like how ribosomes are built and original and functional assays. So this project actually comes from Dr. Schrett's previous work with a previous student that they did a proteomic analysis, and they found this like dysregulation within a lot of mitochondrial genes that are encoded in nucleus, which kind of was a big question mark because literature doesn't talk about that, and there's no clear reason why the defect in the ribosome would cause this dysregulation of the mitochondria. So that's kind of what's led us down this pathway. And then I think finding Dr. Sterling Churchman's work kind of supported this idea that there could be something happening between that.
Danielle, excellent presentation. You're a storyteller, so you should know. This is kind of more like comment we know, but does it matter for you that where the ADP is coming from? Because if you go by total ADP, you cannot actually, right now we do not have it here. So I know that, but does it matter to you that So what is important to you? Total ATP difference, or you really need to identify the source of it, mitochondria or cytoplasm?
I think right now we're just looking at ATP in general, and if our hypotheses are correct, then we should see a significant decrease in ATP in ribosomopathies as a whole, even if we measure the entire cell. And then once we get into more specific assays, we'll do like oxidative stress and stuff that just will focus on mitochondria, to start to look at actually what's happening.
Hey, any other questions? Okay, maybe on the last part.
Are there any drawbacks to using yeast cells instead of like human cells? That like, I mean, other than the fermentation of any source of energy?
Yeah, that's a big one, is that they'll use fermentation and don't tend to do that. Another one is that kind of what we see is their stress, so they'll start to upregulate like their mitochondrial proteins to try and like account for the cytoribosome being lost. Whereas in humans, we see different kind of stress responses happening. And so we're kind of just trying to keep this umbrella term of like we're looking at stress and not necessarily what's happening downstream of that stress, but just that we're able to cause stress or see a stress. So kind of
Thank you. Okay, thank you very much. So welcome back. As the one thing that I failed to say earlier on is we've got the talks broken out into themes. I don't have the themes in front of me, but I think that's kind of why the order is what it is. So the first presenter after lunch is Katie Shields. Katie is a graduate student at Env Health. And I think Katie has kind of an interesting perspective on it because she's got kind of biology and behavior at the same time, which is kind of a pretty neat angle to take on it. It speaks to interdisciplinarity of the health program. So her talk is neural development and cognition in the greater Waxworm. Tell us all about it.
So thank you so much for that introduction. I'm excited to be telling you guys a little bit about my project here with the Env Health program. So to begin with, when we first started this project, we wanted to understand, basically, do moths remember their caterpillar phase? And the short answer is, well, sort of, but first let's back up. The waxworm starts its first active life stage as larvae. In this stage, they eat and grow as much as they can in order to prepare themselves for metamorphosis. Now, what is metamorphosis? Well, this is the inactive life stage that follows, in which the insect's body is almost rebuilt entirely. So what happens if we got this guy? Well, we see a whole lot of liquid. And it isn't until the moth emerges that we see the significant changes that happen within metamorphosis. So to give you guys a little bit of background information, currently research in terms of insect cognition and memory. We know that it implies long-lasting memory after associative learning has been displayed using a classical conditioning olfactory procedure. And also in this tobacco hornworm, which is a type of moth, they have shown associative memory can survive through metamorphosis. So why does any of this matter? Well, our first way of looking at this is pest control. The greater Waxworm is a common pest of honey colonies, so to understand their neural development can help us to target specific stages in development. And additionally to that, we want to look at how it impacts our evolutionary and ecological view, and specifically looking to advance our understanding of how larval experience may impact adult mating choices, habitat selection, and social interactions as well. Additionally to that, we actually don't have a lot of knowledge in terms of insect brain development and what happens during metamorphosis. So this would help us to bridge that gap. So moving on to the first chapter, specifically, we want to look at memory through metamorphosis. That's because the larval peripheral nervous system and central nervous system undergo a considerable amount of degeneration, reorganization, and growth. And specifically, we want to focus on olfactory memory, which is otherwise known as scent. So that brings us to our research question within the first chapter. So does larval memory persist beyond metamorphosis into adulthood? Specifically focusing on waxworm cognition through this major transformational life stage. And what we want to do, our first objective is to train an avoidance response that can survive from the larval stage into adulthood. Now, when it comes to methods, we want to look at classical conditioning, Similarly to how Pavlov trained dogs to salivate just the sound of the bell, we wanted to classically condition odor avoidance within relaxation. Now, you may be wondering how your condition works. Well, I'll break it down for you guys a little bit. So before conditioning, when the organism is presented with a scent, it should be neutral. It's neutral. It should not be aversive or attractive to the organism. So what we do is we pair this neutral scent with shock or an aversive stimulus repeatedly. And then so after we begin to see that they display an aversive reaction to the scent alone. So here's just a little video demonstration of what it looks like in the lab when we shock worms. The black center piece right in the middle there is the gel, which is connected to a power source. And then as well on the left-hand side, that is where the scent is coming in. It's our olfactometer, which just pumps in that ethyl acetate. So they are being exposed to both the scent, and then the shock gets turned on, and this procedure is repeated 6 times over the course of 3 days. Now moving on to our methods when it comes to testing. Specifically looking at moth testing, what we do is we test moths 1 day post-eclosion or 1 day after they emerge, individually in the Y choice olfactometer. So what happens is they are placed in the bottom of the Y stem, and they are given 2 choices: air is being pumped in, control, and on the other side, ethyl acetate. So this air pumps in at 40 milliliters a minute. We have 50 milliliters of the ethyl acetate, and then 50 milliliters of our distilled water. And so they're given about 5 minutes. No, they're given exactly 5 minutes, sorry, in darkness. And at the end of this 5 minutes, we measure where they are within the segment. So moving on to just some preliminary results within the control group. So what we want to understand is this scent neutral, a neutral stimulus that can be used. So far what we found is that they have no preference for the control or the ethyl acetate. It's pretty random and spread across the board, signifying to us that ethyl acetate is a neutral stimulus for wax moths. Now here's just a video of one of our moths from our control group heading up the stem. This kind of just gives you guys an idea of what it looks like when they are in the stem, forced to make a choice. It's on the ethyl acetate side. And we found both in the control groups, they ended up on the ethyl acetate side, as well as in control 2. Now, if we move on to results within the condition groups, we want to understand, does this larval memory persist into adulthood? And so far, what we found is that the wax moths prefer the control or ambient air over the ethyl acetate, and they were avoiding the ethyl acetate. As you can see with the graph here, yellow is the untreated groups, and the blue is the condition groups, and that is their preference for clean air, and shown in a percentage. So we're seeing about 80 to, I think our highest was 87% preference for clean air over ethyl acetate. That's just signifying to us that they are avoiding ethyl acetate and going to the ambient air side. So here's just one of our conditioned moths. It ends up heading to the control side on the left, comes back down to the main stem there, ends up having back to ethyl acetate, kind of freaks out a bit, shows quite a burst of response, comes back down, and heads back to the control side where it spends the remainder of the time in that side of the stem. And this was a very common finding. They kind of displayed that fluttery reaction to the ethyl acetate side, and it just hangs out there for the rest of the time. So if we move on, what is next? Well, these results are preliminary, so we want to continue to condition odor avoidance in the greater Boston. We also want to consider looking at mating. So instead of using a Y olfactometer choice test, we want to look at using a mating choice test. So a conditioned moth would be given 2 mating options: one moth that has been covered in ethyl acetate or lightly brushed, or a moth that has no ethyl acetate. And if we were to do this, this can just tell us that moths, the information there have been learned as, in their larval stage can be applied to a totally different context, such as mating. So to summarize what we know so far, so does larval memory persist beyond metamorphosis into adulthood? And so far, the greater wax moth can remember what was learned as larvae. So we're seeing that the learned odor avoidance does persist through metamorphosis, and that just helps us understand what is really happening to the brain during this stage. So now we move on to our second chapter. Our research question when starting this chapter was, what developmental changes are wax worms going through during metamorphosis? Specifically looking at the pupae stage, and our first objective was just to determine key developmental time points that we wanted to sample. So our methods when approaching this chapter, our first step was to monitor wax rooms every day during patient to determine the average days until emergence under our controlled conditions. Second step was to take pictures every day of the patient during development externally. And then our third step was to dissect every day of the patient and figure out what developmental changes are happening internally. Now, if we move on to some of our results so far, within pupae development, we were able to determine it on average, it takes about 11 days under 24 degrees Celsius with high humidity. These photographs were collected across, they go from days 1 through 11. This just shows the front side of the pupae, and this shows the back side. Now, some key findings from these photographs and dissections collected. On day 1, we're seeing a significant amount of change occurred just within the first day. These 3 pupae are all on day 1 of pupation, and as you can see, the one on the very left almost closely resembles a worm, whereas the other 2 are further along in development. And that just signifies to us that within the first day, they're going through a huge amount of change. Now, if we fast forward to day 6, this is when, on average, you see the first appearance of eye development externally. As you see, the red lines are just pointing to where its eyes are, the pupae. And then if we go forward to day 7, when looking at pupae dissection, we begin to see the eye develop internally. So that red circle is its head region, and the black dot is displaying its eye, and the white stuff there is what we assume to be some sort of neural substrate. So if we go forward a little more, on day 9, we're really seeing the moth adult form take place. We're seeing these compound eyes begin to form. And on day 10, we're just seeing that further development of the adult body. So we're seeing those wings, their legs, and their antenna as well. This makes sense because they're just about ready to emerge. So what is next? Why is this important? Well, we want to target these developmental time points, and we want to use DNA and RNA extraction to identify the genes related to neural development. And this can help us to identify what genes are being expressed throughout metamorphosis and connected back to future behaviors. So to summarize what we know so far, what are the key time points in pupation that we're specifically interested in? Well, day 1, as you're seeing that transformative development from the larval stage into the pupal stage. And on day 6, eye development is only visible externally and not internally. And on day 7, we're seeing that eye development is visible internally. The entire body is still liquid except for its head region has begun to develop. So, what is the use of this research? Well, if we can understand neural development, we can target these critical windows of nervous system development through biological or chemical control interventions, specifically when handling wax rooms as a pest to these honeybee colonies. And furthermore, this can just help us build upon current literature, as we know that we only know that one type of moth has been shown to have memory persistence across metamorphosis. This can add to what is a huge gap within the literature. Specifically looking at insect memory and how if it can persist through this intense neural and motor remodeling, this can help us understand how they choose their times to lay eggs on, as well as mating and feeding behavior too. So I'd just like to take a minute to thank my supervisor, as well as my colleagues in the lab. And that's okay.
Thank you very much, Katie. Hopefully lots of questions from the audience.
So when you're talking about doing like DNA and RNA extractions, will you just take like all that poop out of there? And like, are you assuming that they're all the same cells that in that kind of liquid, or will those be like differential cells?
I guess we don't really have any assumptions about what is happening throughout the 11 days. So we're going to look at taking from every day and basically freeze them, and then we're going to look at the whole body of each organism. So really no hypothesis there yet, but well, thank you.
Just kind of going on with that. So, are you interested in at all in just like including the development line?
Substrate. It is hard to dissecting, figuring out, like as you saw on day 7, that was like the first day you came to see that develop. Whereas like the first 6 days, they're just completely like so can't really differentiate what is what based on dissection. That's why I'm kind of looking at something across the board to get an idea
a baseline, I guess, what does actually happen? Yeah.
Shock was 6 times over 3 days. So just like a second, like just like shock for a second, like how long does that go on?
It's 30 seconds of dragon bolts. A lot of like our initial pilot work was figuring out what can they survive through. So it's two sessions in a day, they're an hour apart. Spaced over 3 days, and there has been like the previous research that was kind of replicating some of those methods. They use dragon bolts, so we kind of use that. We haven't had them die from shocking, so yeah. Yeah. Alright, I'm just wondering, like, I love these stuff, but for quantifying their preference, how exactly do you, like, what is your criteria? Is it like just if they go there, or just beat it for like 5 seconds, or like how do you do that? It's well, right now we're looking at where they end up at the end of 5 minutes, but we're also looking at like pre-record, seeing how much time they spend in both sides of the stem. So it's kind of, you have to just like leave them in darkness, not interfere, and just watch. So that's why the camera set up, and then you measure which side they are on. And sometimes they don't make a choice. That's like when they stay in the center stem. So that happens. But, yeah.
Well done. Okay, our next presenter this afternoon is Jackie Tran and Dr. Carol Rumford's lab. Jackie's presentation is about hatred, pathogen surveillance and management as it relates to talk. So also about it.
Thanks, Mike. So about opinions today. Currently, our amphibian friends are not doing well. They are facing some of the worst ecological disasters in their natural history. Since the 1970s, extinction rates have significantly and steadily increased, and 41% of documented species are at risk of extinction. Some of the drivers behind the extinctions include habitat loss and degradation, which accounts for half of the event. Climate change, which accounts for 15%. Disease, which accounts for 12%. And other factors such as invasive species, unnatural fire regimens, and overexploitation. So as far as diseases go, there are 3 main agents behind this driver: fungal pathogens, viruses, and non-fungal parasites. My research is interested in fungal pathogens, specifically 2 globally emerging pathogens known as EV and ESAL. They affect the skin of amphibians and disrupt vital functions that amphibians need in order to live, such as oxygen exchange, UV protection, etc. And the disease is known as chytridiomycosis. Chytridiomycosis has been labeled the doomsday disease. It has been considered the worst disease ever recorded among vertebrates in terms of biodiversity. And together, both pathogens have caused about the extinction of about around 90 species, critically endangered around 120, and in total have affected around 700 species. So a little bit about the origin and spread. Both pathogens have been genetically traced back to Eastern Asia, their origins. BD was first discovered in Australia following a mass mortality event in the 1990s. B-cell was discovered in Europe following a severe decline in European fire salamander populations in 2013. And their trade and dispersal can be traced back to the 1930s when African claw frogs were internationally traded for biomedical research. So as far as BD funguses go here in North America, they're widespread and abundant. However, we do sit in a data deficient region, both us and Saskatchewan. B-cell, however, has not yet been detected here in Northern America, and their anticipated effects are thought to be a lot worse. So why search for these pathogens here in Manitoba? Well, to protect and conserve our local amphibian populations and the ecosystem services that they provide. 43% of Manitoba's landmass are wetlands, which can serve as highly efficient disease corridors for aquatic pathogens like BD and South. And to fill any knowledge gaps regarding prevalence data and any genetic resistance that our populations may have against these pathogens. So there's been a lot of previous research done on the relationship between both pathogens and amphibians, as well as the relationship between environments and the amphibians, which is very well understood. Only recently has there been emerging literature on the relationships between the environments and the pathogens, and certainly not here in Manitoba. So the aims of my project are to establish baseline data to see the possible effects that the environment can have on the pathogen survival and amphibian immunity, if we were to detect any, and to look at the dispersal pathways at the field sites that we are surveying. So my first hypothesis is that there's significant risk of transmission potential between chytrid pathogens and amphibians with prolonged breeding periods. So BD and USL are aquatic species, aquatic pathogens, so they might share similar hydrological requirements that are that require some level of permanence in their wetlands. Second hypothesis is microclimatic conditions and anthropogenic factors of field sites will influence the detection of chytrid pathogens, and I'll get into that a little bit later. So my objectives are to employ quantitative spatial ecology methods to identify suitability hotspots here in Manitoba, establish baseline field level disease ecology data here in Manitoba, and determine higher priority areas for future conservation work. My first approach is spatial ecology. Enter correlative species distribution model, which is widely used in spatial ecology and conservation biology to model the predicted habitat suitability of the species. Some of the algorithms include, or the methods include generalized linear models, bioclimate, random forest, etc. The species distribution models rely on previously existing occurrence records to plug and ground. For example, here we have one environmental predictor in aperture. So you kind of see species tends to stick around the warm range compared to hot and cold, as determined by the heat map. When I add another predictor in or a variable in, kind of the same story. In this case, mean annual precipitation, and you'll see that the high suitability spots or range will overlap. So that is what the species distribution model learns from. So here I only have two just for explanation purposes, but you use as many as you want to or relevant in terms of environmental predictors. So a bit about the workflow. So just like any other SDM, species distribution models, you need climate data or any predictor variables. I've retrieved climate data, elevation data from the database known as WorldClim, very popular for our standard for species distribution modeling. Presence data, I collected amphibian occurrence data from GBIF, Global Biodiversity Information Facility, and BD&B style geographic occurrence data from a database known as amphibiandisease.org. So the algorithm that I'll be using is called maximum entropy, and maximum entropy or maxent has some unique features to it that accounts for sampling bias. It helps you prevent overfitting and underfitting. I can get into a little bit more about that later, but for time purposes, that's all I'll say. So the output is a map, heat map showing you all of the areas that are similar or identical to the environments that the pathogens have been, or the species have been already found in. So here's an emulated map of Manitoba. And I'll be doing all of this work in R. So I'm expecting to see some overlap in habitat suitability between these pathogens and species that require longer breeding, longer hydro periods to breed. Hydro period is basically just the degree of wetland permanence or water that's been stored at a wetland for an amount of time. My second approach is field work. So I'll also be looking at some site level pathogen predictors such as microclimate, environmental stressors, and dispersal pathways. The hope is to go out every season to account for any seasonal differences in the data. So both pathogens have temperature range from 4 degrees Celsius up to 25 degrees Celsius. So what I'm looking for at my field sites is canopy or variability of structure that might or may not allow for the pathogens to persist. For example, the more variability in the canopy, the more possibility that there are in the thermal exposure spots, which then filters out the available spots for the pathogens to persist in. And I'll be doing a microclimate analysis. The method would be a pinpoint vegetation survey. So I got 3 plots. I got a riparian plot, upland, 2 upland plots. So in addition to collecting plant data at each intersection, which is 40 centimeters apart, at every 2 meters, I'll be collecting temperature and relative humidity data approximately 5 centimeters above the ground surface. Another component of my field work will be looking at the human contributions for the possible ways that humans can contribute at these field sites. So the first way would be looking through any activities that could increase chronic stress in these amphibians, which could dampen their immunity, increasing their susceptibility to disease. And the other way would be to look at any dispersal ways that we can assist in dispersal. So it's through trails, spores can stick on our boots, equipment, etc. So just the literal dispersal of spores, which again can increase the risk of disease. So I'll be collecting some water metrics, pH, dissolved oxygen, salinity data, etc. I'll also be noting proximity to anthropogenic disturbances. And using that data, I'll then try to score each site for the vulnerability of patients. I'll also be collecting biometric data such as wave vent length, which is standard for amphibians. Looking at signs of diseases as well. We'll be collaborating with the SWOOP network of amphibian pathogen surveillance, as well as the Canadian Wildlife Health Cooperative for spotting and lab work. So I'm expecting to see some positive detections at sites that do not have as much variability in their canopy cover, that may allow for more suitable habitat of these pathogens. And I'll also be expecting some presences at sites with significant anthropogenic activity going on. So what's next for me is to filter and extract best info for a Manitoba projection to see where the suitable habitats are here in Manitoba, and then to build a species-specific amphibian species distribution model. For field work, to refine protocols for the next, which is spring and fall 2026. So in summary, K2 pathogens are significant emerging global threats that necessitate cost surveillance everywhere. Presence here in Manitoba is very likely, but we don't know what the infection severity or the amphibian community side of things are. Baseline disease ecology data from species distribution models and field sites can also, that I'm hoping to collect, or that I'm collecting, can inform future conservation efforts of disease management. I just want to acknowledge Dr. Madison, Dr. Beaverton on my committee, and Brandon University Research Committee. Thank you.
So hopefully we'll have some questions from the audience.
I'm just curious. So, what's informed your decision to pick a particular disaster area and October? And also, I just want to know this decision that you're taking, does it kind of fit in to places in terms of parameters or factors that's actually asked this to this maybe universal or the maybe in Australia and
Okay, so transferability. Yeah, so I'll answer the first question. So the reason why is, if you look at the presence points, it's been noted that the occurrence of VD is pretty ubiquitous across Northern America. And so I mentioned earlier too, we sit in a very central spot to these essential spot, which can B-cell isn't here yet. So if we can collect data on VD and figure out the way that it kind of behaves, quotation marks, the ecology data, that can also help us not only prepare for an outbreak of VD, but also in the future event of B-cell. And another reason too is we are an efficient disease corridor. We are full of wetlands. So a lot of habitats that we have here, wetland habitats here, on paper, theoretically are very suitable for this habitat. So in a way, we can almost act as like a, geographically, we might act as a bit enhancements factor to spread. And then to answer your second question, if you're asking about transferability, so the species distribution model that I'm using to build and to identify these local hotspots have already been widely used in the literature. So it's nothing novel. It's not a new approach that I'm taking. Yeah, so it's been done in other places such as Australia. It actually has been done in Australia. Yeah. And yeah, but I hope that answers the question.
Yeah. One last question. Can birds spread it from eating?
Not from eating, but water. It's been observed that waterfowl it can attach to their legs of waterfowl. So that's another dispersal vector.
Okay, thank you very much. Okay, our next presenter today is Toby and Dr. Pete Whittington-Robb. So Toby's presentation is called Modeling Peatland Microtopography in the Sudbury region. So what's your story?
Very much. All right, so today I'd like to take a bit of your time to tell you about the project that I just started working on this last summer, which focuses on peatlands, particularly within the Sudbury region. So just start off, I just kind of want to give a little bit of background about Sudbury itself and some of the unique challenges that it's facing, especially in terms of the peatlands and vegetation within the area. So it is a significant center for both mining and smelting of nickel ore. So in the late 1800s, there was large problems were found as the railway came through. Really, ever since then, especially since the 20s, it's been sort of a huge center for smelting operations, and unfortunately, this has released a significant amount of sulfur dioxide in the atmosphere. So this is how you find peatlands and really the vegetation out. So this is also sort of a lot of harvesting of trees, particularly within the area, be that those smelting operations, well, just for other applications and stuff. So it's really had a dramatic impact on the vegetation, the trees, basic peatlands, everything involved in that. Sulfur dioxide inhibits sort of nutrient uptake. This is that is sort of critical components of the function of vegetation. So in the 1970s, it was the construction of the Eco Superstack, which was one of the largest, it was the largest chimney at the time, basically to release some of these pollutants further up into the atmosphere. So the added the solution really they were just trying to get it higher up, get it further away and spread it over a larger area. So some of those impacts weren't impacting the entire region so much. So this was kind of seen as a beginning of steps to kind of be able to remediate the area because so much sulfur dioxide was affecting these plants. So as you can see, there was over 82,000 hectares that were basically described as being almost completely barren. There was almost no vegetation in the area whatsoever. But sort of using lining efforts and different seeding, they were actually able to remediate a significant portion of that. So sort of normalized it in a sense to a degree. So as you can see, you would almost not notice that at all today, looking at sort of this peatland here. So really it's kind of a success story, right? So we have this nice forested area, the cityscape and the water, right? But really what we're trying to look at here is how exactly is that still impacting the peatlands within this area, right? So the main three purposes and objectives of what I'm trying to do here is sort of quantify the degree which peatland disturbance and recovery can be correlated with microtopography. So really just sort of how that sulfur dioxide is impacting some of the smaller scale landforms within the area. Calculate carbon storage, which is essentially the loss to disturbance. And lastly, to better understand how accurately the models we're using can represent the microclimate, right? So it's important to know that what we're using to determine these other two factors is actually realistically representing what's going on at the surface. So to start out what exactly a peatland is, it sort of has three core things that involve in its development and existence over time, and that being sort of consistent saturated water levels, organic materials, and then the passage of a lot of time. So that eventually leads to development of peatland. So look at that process as a whole. We might have an area which sort of has bedrock or something underneath, so we get consistent water levels. Vegetation, organic material dies, it goes into the water, it can't decay fully because the water is sort of a living factor, there's only so much oxygen. That leads to these development of these thick layers of peat over time and storage really of a significant amount of carbon. So what we're looking at is kind of like the small scale features within these peatlands. So it's often referred to as the microtopography. When I kind of reference microtopography, I'm talking specifically about these hummock and hollow features. So hummocks are kind of like these little mound structures. They're about 1/2 meter to about 3/4 of a meter usually. And they're just like this mound-like elevations within the landscape. And then you have these hollow features, which are just like these depressions kind of situated adjacent to them. So you just kind of get this undulated water feed up and down, up into the hummocks, down into the. So just kind of giving you an idea of what this looks like. Sort of a real surface, right? So this is definitely really exaggerated, but gives you an idea, right? So these hummock formations, you have these nice sort of rising up within the landscape, accumulations of peat, and then the hollows, these lower lying depressions situated adjacent to them that are much more closer to the water table. So just kind of giving you a more typical example. This is kind of one of the sites out in there, right? So we can still see some of the hollows, right? So you see hummock here down into a hollow, hummock, hollow, right? So we still get that undulating topography, but not as pronounced kind of in the previous section. Still definitely visible within the landscape though. So why do we care about these microtopography, hummock and hollow features, right? So they've definitely been correlated with peatland resilience. So they're very important for some of the positive feedback loops that go on. In order for peatlands to form in the first place, as well as it exists for a long time within the landscape. Carbon storage, biodiversity, so certain species have been associated with hummocks and hollows, so amphibians, reptiles, moss species that are associated with hollows respectively. And then lastly, this is kind of isn't within the literature, but sort of a hypothesis of people who have spent a long time within these landscapes is that they could possibly be an indicator of disturbance zones, which leads kind of like the main focus or question of what we're trying to look at in our project here. So are these peatlands flattening due to reduction in moss caused by the smelting operations, right? So is this sulfur dioxide inhibiting moss growth enough to, rather than having these significant undulated topography, you kind of see a bit of a flattening of the so looking at the region in particular, so you see these red areas are the smelting operation. So we tried to pick sort of 5 sites that exist kind of within a gradient of what's the ideal disturbance would be, right? So areas which are located directly beside one, right down kind of the wind path, right? So sort of our most ideally disturbed ones that are in sort of moderate proximity, not directly in the wind path. Winds are westerly, west to east, so maybe sort of moderately, and the one up here, so on the opposite side of the city and furthest away, right? So our minimally impacted site. How are we going to do this? So we're going to be using drones to collect imagery. So we have the Mavic 3M here. It's capable of seeing the visible spectrum, so red, green, blue, or those sorts of things that our eyes can see, but also able to see in the near infrared and red edge. So either sort of parts that can be important for looking at plant health, but might sort of give a different view or create different lesion surface models for us. It's used in tandem with the RTK or real-time kinematic thing. It's We don't know anything about serving it. Basically, it's almost a base station, but not quite. It just sits at a location, detects errors within the GPS signal. It basically tells the drone what the errors are. So when it's flying around the landscape, it's much more accurately knows where it is and just leads to much more accurate models. So we'll go to these peatlands, do a flight route, the north-south pass. It will also do the east-west pass. When the need flies or reaches a particular station, it will take imagery straight down as well as oblique imagery, which is imagery that is not it's at a bit of an angle, so 27 degrees. So as you can see, it kind of looks back, looks down, looks forward, looks back. So this just really allows us to create much more accurate models as well as a significant time saving feature. So what type of models am I talking about in particular? So we're using digital surface models, which is a 3D representation of Earth's surface that basically contains all the vegetation, all the built-up features are part of the model. There's no masking techniques going on. So it's derived from a point cloud, which is produced using that aerial imagery. And it's typically displayed in a raster format with every cell representing an elevation. So it's almost kind of like looking at like a TV screen, you pixel holes of information, put them all together, and it kind of gives a bit of information on the surface. All right, so to drive it home, this is the peatland. We do our drone flights. We build our digital surface model, and ultimately, is to build a mosaic, which is just a stitching together of the photos that go into creating that digital surface model to just allow for the analysis. It'd be more easy for us to determine where exactly these transects are, which I'll kind of show you in a second here. All right, so looking at this digital surface model, this was the picture I showed you earlier, looking out into the peatland, these hummocks and halls I pointed out. These are kind of those ones right here, right? So we know that these digital surface models are capable of showing us these hummock and hollow features, but then the question comes, how accurate, right? Is it actually telling us what's going on the surface, and can we know that that's actually true to what we would see? So that's where the ground truthing component comes in, right? So it's just our attempt to compare what we see in a digital surface model versus those sort of measurements that we'd actually get if we went out to the surface and just know that the digital surface model is telling us the truth. So we go out to our peatland, put 2 stakes down for our transect. Use a water altimeter to measure exactly where the ground is, as well as how high the vegetation is compared to the ground. And really, we just go along at regular intervals, whether it be 10 centimeters, 20 centimeters, we kind of take those measurements so we know exactly what a transect should be telling us when we get that data out of the digital surface model. These also have mirrors attached to them when we do the flight, so that way we know exactly in the imagery, those orthomosaics, where these transects exist. All right, so this is just kind of what it looks like in practice. And here, if you can see, right, so we see these little mirrors within the imagery, okay, we know we're both on mosaic. So we know where that transect exists in the digital surface model. We have measurements along those regular intervals. So we should be able to accurately compare the digital surface model versus what surface actually is showing us. So this is some just extremely preliminary results. This was the transect that I just showed you. But so that you can see that so the black is the ground, the green is the vegetation measurements that we took, and then the orange is what the digital surface model is telling us, right? So there's pretty good correlation with sort of the movements, right? So we know that it's really accurately measuring the vegetation, but one of the key things takeaways for us is that it doesn't really detect the ground ever, in terms of even if we might say there's like low vegetation here or low vegetation here, it almost never detects the surface of the ground. It's always interpreting the vegetation as surface, if you understand what I'm saying. And then another one here, so that one was transplant, which was one of the most impacted. And this is South Cartier. So this one was taking at a little lower resolution, so longer distances in between our transects. So we'll probably have to go back and do it again for, I think, to capture one of the lower end of sort of a higher resolution. But we can see that for a lot of areas, it's almost within a centimeter to kind of capturing that variance of topography, but once again, not really showing the ground. So it's important for us to know that therefore that the model is basically always interpreting the top of vegetation as the ground. So next step, so basically you conduct these accuracy assessments for all of them, so both for all the digital surface models against the parent site. 5 different sites, 6 different sets of parent sites for every site. And then as I talked about the multispectral imagery, so introducing some of that, we might get some different results. So just in summary real quick, so these peatlands are significantly impacted by the sulfur dioxide released from the smelting operations. Likely led to, or it has led to a reduction in moss growth, which could potentially begin to apply on the surface. And what we're trying to use is these digital surface models to determine what degree they've been impacted, and if we can use this over a larger scale, sort of study different impacts on all the peatlands as a whole. My references, just thank you to especially Dr. Whittington for coming out to the Univ field a lot of days, and all the people who funded me, and yeah.
Okay, hopefully we have lots of questions.
Nice.
So, how does the camera figure out the elevation of it? In some way, looking at some sort of discontinuity, or is it the same signal as
It's basically the concept of the parallax shift. So because we have so much overlap in the imagery, it detects the amount of shift basically from the focal point of the camera. So as you're kind of driving through space, it's almost like that effect is, you know, if you're driving down the highway and something close to you, it moves a lot faster, whereas it's further away, it moves a lot less. So the camera basically detects that and can relationally tell where something is in the landscape. Right? Yep, that's right. That's it.
It's also got a real-time kinematic, so there's a base station set up, so it's getting like centimeter level accuracy GPS data in live time, so it also knows where it is in the sky as well.
And so, where in your matching up your DSM to your vegetation on your ground, so where the DSM drops down from the vegetation to the ground, is there a gap in the vegetation at that point?
Well, that's the thing, is the digital surface model almost never detects the ground. So there will be, we notice, I mean, it's extremely preliminary, maybe like a slight drop.
There's actually a slight mislabeling of this. The DSM is the green and the vegetation is the butter. I've had it between last week and today.
I got it switched. Oh my god.
So the green line is what the model is seeing.
Yeah, sorry, that's my apologies. So yeah, so this, so the green is in this labeling. So the vegetation basically always, even if we kind of, I mean, that's the thing. It's like, what exactly do you call no vegetation, right? So it's like 3-stranded grass, low vegetation, like, so there's kind of like an involved into it. So we were kind of, I mean, I was surprised that what we determined the vegetation was so close. But yeah, like, so apologies for that, but like basically it's never detecting the ground as far as we can tell. But that might change, kind of like I said, introducing maybe the multispectral imagery, maybe using some different interpolation in the surface model production. We might, or looking at the point file itself. But I kind of mentioned we might actually be able to determine where that ground actually is. But yeah, sorry.
We have time for one last question.
Is research like this something that the mining companies support, or is it something that the mining companies don't want?
Well, I mean, I don't think they would be overly against it, because I mean, there's definitely no secret at this point that it caused significant damage. I mean, there's so many regulations and stuff that have been put in place in terms of capturing or, you know, like reducing the amount of emissions that they've done as a whole. So I think the damage is kind of done at this point, and you might be able to kind of chime in, but I don't think there's really any pushback against it from them, because they already have all these other techniques which they by law have put in place to reduce their emissions. So it's kind of just like doesn't really matter to them as far as I know.
It's a good question. So, I mean, the funding that we got for this was not specifically that there's no money in the budget to do that, but when you get these large NSERC grants, there's sort of economies of scale, so you can take other little pots of money and then do more like discovery-based research at the same time. So we'll present these results and show them to industry, right? But I mean, the impacts on the peatlands are well known, we're just trying to quantify them so that we can. You know, have an assessment of what's been done and then target peatlands for restoration, like we're applying for another. Series of grants based on some of this information. As well, on like what peatlands would be best to target for restoration efforts, so from. The industry's point of view, it's probably better bang for their buck if we can target peatlands that are. More restorable, or have a higher restorability, because they already have some of this. Like those sorts of things, so it all feeds back in, so. It's a good question, thank you.
Okay, thank you. So last but not least today, we have Cole from Dr. P. Berman Research Group. And Cole is going to give us a presentation on alignment analysis and alteration mapping as a vector for IOCG style mineralization. So tell us all about that story.
Thank you. So first, I kind of wanted to start off with some of the big picture objectives for this project. So hope to assist in exploration activities at the Cancer District to try and find critical minerals and precious metals, as well as improving the overall exploration model for the area, and in turn kind of reducing the cost and environmental impacts of doing exploration for these deposits. So why do we care? Well, IOCGs form some of the biggest deposits on the planet. A good example is the Olympic Dam in Australia, which forms not only the world's single largest source of uranium, but it is also the largest mineral deposit on the planet, as last year having a resource of almost 10 billion tons. So they can form very significant reserves of critical minerals and precious metals, and it's a very long list of things that can come from these deposits. So what is an IOCG? Well, iron oxide copper gold deposits are systems that form in the guts of volcanoes, essentially just above the underlying magma source, which is represented by the orange area here. So your magma exhales hot volcanic fluids and gases, which alters the rock surrounding it and causes the deposition of abundant hydrothermal iron oxides in the form of both magnetite and hematite, shown by the black and brown areas here. And it also causes a lot of alteration and recrystallization of the wall rocks surrounding the deposit, giving you a very spatially extensive, intense, and diagnostic alteration halo. In the highest temperature zone near the magma, you get sodic alteration, and as you get further away from the deposit, you get sodic calcic, high temperature potassic, and the low temperature potassic alteration. And at the very external part of the system, represented by these orange lines here, you get epithermal veining, which would be an important invader. So these deposits form in volcanic belts, so that makes them very heavily structurally controlled from the regional scale all the way down to the microfeatures. So the area that we're doing exploration for these deposits is the Great Bear Magmatic Zone, which is essentially a 2 billion year old Andean style volcanic belt that's exposed on the northwestern portion of the Canadian Shield. It's exposed from about the northern arm of Great Slave Lake to the east arm of Great Bear Lake, and continues under sedimentary cover both to the north and south. So the belt is dominated by granitoid batholiths that post-date magmatic and hydrothermal activity in the area. These are represented by the area shown in pink, and the remainder of the belt, shown by the green areas, represents volcanic rocks that show extensive hydrothermal activity and mineralization, and are the target of our exploration throughout the belt. So to date, there have been 10 mines that have produced throughout the belt, all of which are no longer producing, and all of them produced from epithermal veining, so just that very superficial portion of the environment. And now currently there are 2 IOCG style deposits that are under development, both in the very southern portion of the belt, the Eco and the Sudan. So the area that I'm working in is the Council River District, which is kind of in the north central region of the belt. So Council River has 4 past producing mines that operated between 69 and 85, all of which primarily produced silver with a little bit of copper and a byproduct cobalt and bismuth.
So at some point, there were some internal reports found from the mines that indicated there was also some gold produced. It was never reported to shareholders, so the amount isn't actually known. But with the price of gold skyrocketing over the last 5 years, this is what brought the interest to do exploration in the region here again. And keeping in theme with the Great Bear, everything previously mined through this district was all from the epithermal veining. So it wasn't any IOCG related deposits. Kind of when the mining activities were winding down in the area, the Geologic Survey of Canada decided to run a regional mapping program where they unraveled the whole volcanic history of the area, discovering what's known as the Black Bear Collapse Caldera, which is essentially a 20-kilometer diameter collapse of volcanic caldera that's been filled with various pyroclastics and volcanic rocks. At some point, the property was also intruded by the Rainy Lake Pluton and the Balachi Plutons, and currently it's thought that there is a compositionally related body underlying the cauldron, which would have provided a heat source to drive that large and strong hydrothermal alteration system that's affected by the rocks in the cauldron. So when the geologists from the Geologic Survey were mapping this area, they noted locally infrequent, intense alteration throughout the property, but didn't relate it to an IOCG style system because they simply just weren't discovered in the 80s yet. They eventually got discovered in 1992, and then over the last 25 years or so, with work by the Geologic Survey of Canada and many others, the IOCG potential of the property was discovered. So here's an air photo of the property, kind of just showing the areas of past production. The Terra mine was where the vast majority of silver production was, with Norex operated as a satellite deposit to Terra. Rain Vein is just a minor offshoot in Norex, and the North Rim and Smallwood mines just having very small amounts of silver production. So that brings me to what am I doing here? Well, there's a lot of things to kind of look at with a project like this. Well, one of the things I hope to define well is the mineralogy and the chemistry of the distinct alteration phases that are appearing on the property, as well as determining what kinds of mineralization are present and how they relate to the different styles of alteration on the property. And determine if the Balachi Pluton and Rainy Lake intrusives actually are related to each other, and if they are, what their relationship is to the alteration, mineralization, and volcanism on the property. And finally, kind of take all of the things that we see and try and draw comparisons between this district and other districts throughout the belt. So the first thing that I did here before going up to the first field season for mapping was a lineament analysis. So you might be wondering, what is a lineament? Well, a lineament is essentially a linear feature that can be interpreted from various types of aerial imagery, like digital elevation models or topographical aerial photos. So just to kind of give an example, you might, there's some discrete outcrops shown here in the photo, and you might interpret this linear feature splitting these two in half to be either a fault or a lithological boundary. So generally in the Canadian Shield, you have excellent correlation between topography and geologic features, and in most cases also structural features. Mainly this is due to differential chemical weathering and glacial erosion of areas of contrasting tectonic and lithological competencies. So essentially these lineaments can be used as proxies for structural features. And since we're operating in a volcanic belt, it's also important to try and interpret ring structures, which can indicate domal features, or collapse structures like the Black Bear Cauldron. And as we saw in the photo of the property, there's fairly good outcrop exposure, which allows for particularly good correlation. So here's the lineament analysis done on the property. I used a subjective classification of lineaments just based on relative visual intensities, with primary being shown in red, secondaries as orange, and tertiaries as yellow. The only thing I want to point out on here is that the main structural theme that you see in the Great Bear magmatic zone can be seen in the property again here with a series of northeast trending features that cut and offset these southeast trending volcanic belts. So the first season of field mapping is done this summer. All of it's being done electronically with QGIS. You have all your data geo-referenced. Since this is an alteration project, more or less focusing on mapping the distinct alteration zones. But with geologic mapping also comes many other things as well to consider, like rock types, mineralization, and structural features. So the area that was mapped this summer is shown by the dashed red line with plans to expand to the northwest and the southwest for next summer. Now let's we've done the first field mapping season, we can start with some of the analytical work, which all starts with thin sections. So a thin section is essentially just a 30 micron thick slice of rock that you can analyze under a microscope. So for the most part, we'll be using reflected and transmitted microscopy to try and determine the mineralogy of the distinct alteration cases on the property. We'll also be using the scanning electron microscope and X-ray diffraction machines as well. The SEM for the most part will be used for determining the nature of precious metals in some select samples, but will also probably be used for determining some unknown phases as well. And the XRD machine primarily is going to be used for mineral identification. The next thing that we use a bit is some geochemistry. So this starts with the careful selection of what's interpreted to be the least altered versions of each rock type on the property. And then you can take variously altered versions of each of those rock types and do whole rock and trace element analysis. And in the end, we use mass balance to try and determine how the rocks have been changed chemically and try and relate those to the mineralization of the property. The last thing you might do with this is build a residual alteration index plot like the one shown here, which essentially just gives you some of the ideas of what the regional hydrothermal patterns look like. And the last thing that I hope to do with this is some geochronology, which is going to be just argon isotope data. This will be done to determine the timing of some events that occurred on the property, like altering events, mineralizing events, and intrusive events. So just kind of summarize what I hope to accomplish with this project, like to examine the mineralogy and chemistry of the variously altered rocks on the property and try and determine the relationship to mineralization, as well as examine all the different styles of mineralization and try and determine how each of those relate to the distinct alteration cases found. We'll also like to use age dating methods to try and determine the timing of various events that happen on the property and take the results and try and make comparisons with other districts within the belt and use all that in both to try and improve the exploration model for the Amazon River District. And I'd just like to say thank you for all the supporters of this project, especially Demco, the owners of the property, for the opportunity to work on such an amazing project, and my supervisor, Dr. Moonman, for the mentorship and support throughout the project as well.
People. Hopefully we have another really good round of questions.
So for your smear analysis and thin sections of your chemistry, are these surface samples you're collecting?
Yeah, all surface samples.
Okay, and then I guess the idea is that it should be the same below.
Yeah, for some distance. Yeah.
Do you have a sense about how far down you extrapolate down?
Well, it would depend on the structural nature of the rocks. If they're dipping, then it's not going to be continuous for some distance. If they're laying flat, then it would be continuous for some distance. But I don't know a pile about the structural aspects of the property. The area is it's not super deformed, but there's still a lot going on.
Yeah, so is the idea that this can be then to maybe guide some more detailed exploration and drilling and things like that, or what's?
Yeah, basically trying to vector into good places to drill, because even just to get a drill up to that location, it's so remote, you need like $3 million to think about drilling. up
Yeah,
there.
yeah.
So essentially we wanna try and do as much as we can before doing that.
It's a road access.
No, the closest road is 400 kilometers away.
Okay.
Other ice road?
The closest road is an ice road. Yeah. The closest paved road would be like 50 kilometers west of Yellowknife. So that's why it's so expensive.
The fun going up there, like it's somewhere that's like pretty remote. Kind of like rugged.
It was pretty fun. It was a good experience. I don't want to complain, but the black flies were horrendous.
Yeah.
They sure tried to carry me away.
So how does one get there? There's no roads. Not really sure.
Well, we had to take a twin otter from Yellowknife, just a small passing point. And then they've got an airstrip there.
So fun questions are always great, but first question.
I was just curious whether some of the mining starts with eight up there.
It's, well, no, it's been abandoned since '85. This company actually that owns this was in this pretty shady practices. That's why it was never reported to their bills or anything like that. So it's essentially just been abandoned and it's a mess. Okay, like the government is working on doing some remediation stuff right now. I think they just kind of started cleaning up the terra mine site this summer. And then they're gonna, I don't know what their timeline is for that, but yeah, it's a pretty big mess up there right now.
Maybe one last question. Seeing none? Okay, thank you. So this brings us to the end of an absolutely fantastic day of talk. So thank you to the presenters and thank you to our organizers. And thank you to the audience for coming up with such really good questions.